So welcome everyone, both here, elsewhere in the US and abroad, to the seminar for the Photosynthetic Antenna Research Seminar Series. It's my pleasure to introduce today Dr. Melih Sener from the Theoretical and Computational Biophysics Group at the University of Illinois. This was a group that's part of a National Institutes of Health resource for molecular modeling and bioinformatics that was started by Klaus Scholten in 1989. It's been a very productive group there at the Beckman Institute. Melih received his undergraduate degree from physics and mathematics in 1994 from the Middle East Technical University. He then, in 1999, earned his PhD in physics from the University from physics at the State University in New York, in Stony Brook. And then in 1999, he went to work as a postdoc with Klaus Scholten up in Illinois. In 2005 to 2007, he moved to Cornell to do work there in the medical school. And then in 2008, he moved back to Illinois, working where he is now collaborating in the Scholten group. He comes from a very theoretical physics and mathematical background, but his interests are broadly in energy transfer and structure and photosynthesis, particularly in the assembly and dynamics in the multi-protein complexes, which comprise the reaction to the light harvesting system, and then funneling that energy to reaction centers. He has another broad range of interest in that general area of macromolecular structure and dynamics. But today he's going to talk to us, I think, broadly about his work in photosynthesis. So Melih, thank you and welcome.
Thank you, Dewey. It's actually a pleasure to be back here. We have a lovely campus, and back in Illinois we don't have much of geography, so I appreciate coming back here every time. So I have to admit that the title is a little bit overly ambitious, perhaps. I will talk about some physical principles of some photosynthetic systems. So most of what I will talk about will be representative rather than exhaustive. But I hope that I can tell you in some greater detail, especially the work that we have done on the purple-bacterial chromatophore. Before I even get started, I'd like to acknowledge my collaborators. Firstly, of course, I've been working with Klaus Scholten for many years, and he's been my mentor and colleague. And I would like to specifically acknowledge actually 3 graduate students that I had the privilege of working with: Johan Strumpfer, Jen Sin, and Daniel Chandler. Danielle and Jen actually recently got their degrees, and I will specifically mention their work as I go over them. And it is not too much of an exaggeration for me to say actually virtually all the recent work that we have done on the chromatophore has been based on the collaboration that we have had with Neil Hunter's lab in the University of Sheffield. So I'm sort of waving at them if any of them are watching. Recently, I had the pleasure also working with the labs of R.V. Freiberg, Chris Schippo, and Simon Shoring, and I will mention some of our work later on. So just to set the stage, what I intend to do right now is to tell you a little bit about the basic physics of light harvesting. So I beg the patience of the experts while I do that, but I think it's sort of a necessary staging ground for what has to come afterward. After that, I will talk about sort of the structural organizational principles as well as how we construct models for supramolecular organizations that are used in photosynthetic light harvesting. After that, I would like to sort of switch gears a little bit and try and see if we can gather any lessons that are sort of physics-based organizational principles that likely have shaped the evolution of photosynthetic species, because some of the features appear as uniform themes across multiple light harvesting mechanisms. So maybe nature is trying to tell us something. So with that note, let me tell you my intention in a little bit more detail. So I will talk about a number of principles such as the optimality, robustness, reliability, and the adaptiveness of the systems that we study regarding light harvesting. Now, the terminology here is, of course, liberally borrowed from the parlance of control theory. And this dates back several decades to the works of Prigozhin and other people. So this is not exactly a new attitude towards studying biological systems, but it's perhaps a language that we don't use nearly as frequently. Now, optimality refers to maximizing some kind of performance. Now, the trouble, of course, is that what do you mean by maximizing, and how do you define performance? This is, of course, a rather touchy issue. By the way, one disclaimer before I go further. When I say design principles of light harvesting systems, what I really mean is physics-based design principles that govern the evolution of light harvesting systems. So there will be no discussion of theology today, just in case we got sidetracked. And further down, just as a note to the complexity of the subject, Bob had this recent paper with a who is who list in photosynthesis, and as I understand the story of this paper, there was a very long discussion in trying to define what efficiency of a photosynthetic system is, specifically in this context, comparing photovoltaic versus naturally occurring light harvesting systems. And even for a basic question such as what we mean by efficiency, the discussion that we have to go through is actually substantial. So instead of trying to be comprehensive, and I refer to optimality in the rest of this talk, it will be more often than not be a specific optimality of a specific sub-process. This has much more limited focus, but it is more easily quantitatively tractable across multiple systems. And I will give several different examples from different processes. And maybe we can have a discussion, I'm hoping later on, to have sort of an overall economical view of the light harvesting process in terms of what it tries to optimize. This is, of course, a greater bearing on developing biohybrids and biology-inspired systems as well. Now, the other concepts of interest to us is how the system deals with problems. On one hand, you have the robustness of the system, which essentially is the ability of the system to deal with immediate disturbances and uncertainties. The typical example of that would be, for example, thermal fluctuations. So this requires a certain parameter in sensitivity in the dynamics that governs the light harvesting process. The next concept of relevance is reliability, which essentially is how does the system deal with failure of components. And so this brings us to graceful degradation. What do you do when one of your subsystems fail? How do you handle it? And the last one then is that of adaptiveness. The example that is in mind is that a light harvesting system will grow a completely different solution depending on which environment it was brought up in. I mean, examples are many. If I were to pick one out of my pocket, it would be, for example, in iron deficiency cases, photosystem 1 develops islands of light harvesting complexes around itself, and if you don't have iron deficiency, you don't have such a problem. So there are problems at protein level, there are problems at system level, there are different manifestations of adaptiveness that one can actually find in light harvesting systems. One note, the last three concepts, robustness, reliability, and adaptiveness, in my opinion, they're sort of bundled together in a natural way because they are all a measure of the ability of the system to respond to problems sorted according to time scale. So if I were to go by analogy with a human being, for example, I would call you robust if, I mean, in this parlance again, I'd call you robust if you can deal with your day-to-day issues, like, you know, you have immediate little problems that get into your way as you're trying to handle your little things, and you have the ability to handle them. I would call you reliable if an emergency happens that will ruin your day, but you can still deliver what you can and what you have to at the end of the day. I'd call you adaptive if you make life-changing decisions that over a long time put you on the kind of course that actually gets you to succeed in whichever process that you were undertaking. So I think there's sort of a natural separation of time scales, and of course, in the context of photosynthesis, the time scales in question will be, you'll be dealing with picoseconds here, you'll be dealing with maybe the life cycle of a protein, and here you'll be dealing with the life cycle of an organelle or a cell. And so that will be the general scheme that I will keep in mind. I will now put this aside, and I will not come back to this till the latter end of my talk. but I promise to give you some quantitative examples later on. So first, let us take a step back and have a look at the physics of the light harvesting process. The first, more substantial step, of course, is the absorption of a photon, and then the deliverance of the energy of the absorbed photon to a reaction center. This is originally the work of Forster that describes essentially the geometry of the pigment network in terms of interactions that govern the excitation transfer process along this network of pigments. So in other words, pigment geometry determines an effective Hamiltonian. The effect of the Hamiltonian on the one hand gives you spectral information. So essentially the eigenstates of the Hamiltonian have something to do with the spectral response of the system without getting into detail. On the other hand, the Hamiltonian also governs the excitation migration process. So with this, for example, you can consider a picture of the extonic structure of a light harvesting complex. I will pick examples dear to my heart. So this is a reaction center light harvesting one complex. It could be in monomeric or dimeric forms, depending on which species. And so here, for example, you see a gallery of the lowest lying eigenstates of the relevant Hamiltonians that are given by the pigments. Once you do this exercise, then you can, for example, consider transfer of excitation between one system and another. You can compare your results directly to the spectroscopic observations of these systems. And so this way you have a direct means of comparing structure to function through the use of an effective Hamiltonian formulation. In this particular example, for example, you can see that the monomeric versus dimeric forms of reaction center LH1 complexes actually have fairly similar response times for excitation transfer, also actually comparable light harvesting profiles, so that probably doesn't come as a surprise. Which brings out the question, why do some species actually develop dimeric forms as opposed to monomeric? Turns out that this is not a question that you can actually answer by the kind of rationale that you can get from the perspective of excitation transfer only. So one important bit about purple bacterial systems, this is in contrast to, for example, a chlorosomal system. In purple bacteria, there is a natural hierarchy of pigment groups. And it turns out that nature is at least kind to us physicists. Whether it actually has a functional role is a separate question, but it is very useful for us for the following reason. You have ring-shaped clusters. So this could be in the peripheral light harvesting complex LH2 or the LH1 which surrounds the reaction center. But what you have is closely packed clusters of pigments that immediately delocalize the excitation that they absorb. And then the transfer between these separate clusters of pigments can be described in the language of generalized Forster theory. And so the description then is that you have a strongly coupled donor transferring energy to a strongly coupled acceptor, whereas the individual, I should probably not get into the screen, whereas the individual couplings between the pigments of the two groups are weak. And it turns out that if you try to do that sort of a description, for example, for a chlorosomal system, you would fail because you have a continuum of strongly coupled pigments over macroscopic distances, which is essentially the reason why today nobody can quite adequately compute the chlorosomal excitation transfer characteristics. I mean, you can hand wave in various formulations, but none of the methodology that has been successful for purple bacteria systems would work with the same kind of confidence. Now, one important note, there's an underlying assumption when you try to describe the light harvesting, the excitation transfer characteristics in the modified Forster formalism. You essentially assume that before the transfer from the donor cluster to the acceptor cluster happens, the excitation distributes itself according to a Boltzmann, distribution. And This is not obvious. So I'd like to, at this point, acknowledge actually the work of Johann Strumpfer, a graduate student in our lab. Something that he did was to develop a hierarchy equation-based method for the time-dependent computation of the density matrix. And what he found out was that in less than about 1/10 of a picosecond, the excitation equilibrates and the extonic levels actually distribute themselves in a Boltzmann-like manner extremely rapidly. So just to give you an idea, if I have two LH2 rings, the 850 rings, trying to transfer energy between them, the time scale for that, if I'm placing my LH2s next to one another, is on the order of 5 to 10 picoseconds. Here you have 1/10 of a picosecond time nearly for the equilibration of the energy. So what this shows that within the approximation scheme that you're intending, the generalized first order formalism works like a charm. So essentially, I repeat again, we got lucky because the first time we studied these systems, which I believe was in 2007, we are dealing with a pigment array of about 4,000 bacterial chlorophylls, and there is no way you can treat a 4,000 bacterial chlorophyll system with the kind of coherent formalism that you would like to throw at a strongly coupled pigment group. So a hierarchy of pigment groups distributing themselves into separate strongly coupled clusters is sort of a boon for us. So one note as I sort of move toward the description of entire chromatophores is that photosynthesis actually integrates multiple time and length scales by its very nature. I mean, you have to deal on the one hand with the quantum mechanics of individual pigments. On the other hand, there is a supramolecular organization that takes place at length scales of about 100 nanometer, as well as you go into problems like assembly, the time scales relevant start to climb towards seconds and then minutes. And then in fact, as I'm told by my colleagues in Sheffield, the average lifetime of a chromatophore vesicle is probably longer than that of a bacterial life cycle. So that daughter cells usually actually inherit intact vesicles from their parent cells. So there's a wide variety of time scales and length scales. Naturally also, therefore, there's a wide variety of theoretical methods that you have to employ for the study of these systems, and there's a corresponding diversity in the experimental methods that are necessary. So basically, this is going to keep us busy for a long time. So a bird's eye view of a chromatophore vesicle then is, it's a pigment protein, it's a supramolecular assembly of pigment protein complexes and supporting proteins in a pseudospherical arrangement, typically with a diameter of somewhere between 50 to 70 nanometers. And in this example, you see over 100 proteins. Most of them are LH2s, actually. And LH1 reaction center complexes form dimer arrays. So these are in red and blue. And you can also see about half a dozen BC1 complexes posted into the mix here, as well as one ATP synthase. The function of the system can be summarized as a diagram where you absorb light by the light harvesting proteins, and the excitation energy moves to the reaction center. There is a charge separation event which is repeated twice, followed by the diffusion of a charge carrier molecule quinone into a BC1 complex. Then the same electrons are actually shuttled back by a cytochrome C2, and the BC1 complex sort of does a sleight of hand for the charge gradient, replacing it for a proton gradient, and the proton gradient, the proton motive force that results, then drives the ATP synthesis at the ATP synthase. So you have one closed box. It eats light, spits out ATP.
Well, it's now clear that structures like what you've shown here that are sealed vesicles really do exist.
That is a tricky question for several reasons.
I mean, I know Neil says this, but...
To the best of my knowledge, there is still some dispute, because EM images, as best as I'm aware, show vesicles that seem to be detached, as well as vesicles that are actually still attached to the cytoplasmic membrane. we have considered both possibilities. From the perspective of studying excitation migration, this is of course not important. The reason why this would be particularly important is when you get to the topology of the protomotive force and the way it is handled. I mean, if you consider the possibility that you have a closed vesicle, and let's say for some reason the vesicle assembled without an ATP synthase, then you have a vesicle that is dead on arrival. In a similar way, let's say you have a vesicle, I mean, I said that these things live a long time compared to the lifetime of the cell itself. If for some reason the ATP synthase becomes dysfunctional again, the entire vesicle will fail. I will mention this a little bit further on, but I want to say at this point that this picture is a lie for a very specific reason. When I talked to Bob Niederman, who has been growing these vesicles for a very long time, he will tell me through his gel electrophoresis experiments that there is content that has never been imaged by AFM or other methods. And there is nearly 200 different peptide types, not proteins, but different types of peptides that he detects in vesicles that have been grown at different conditions. And some of which, for example, includes ribosomal content. So there are probably repair mechanisms, there are probably other adaptation mechanisms that we are not yet familiar with. None of this is accounted here. The nice published images of vesicles are almost always sort of neatly arrayed LH2 domains or LH1 reaction center dimer arrays. And in fact, so far, there are no images of BC1 complexes or ATP synthase either. How this was constructed is a long story. I don't want to get into the details of it, but let's just say that there's a consensus model based on talking to the experts. That's really sort of the skinny of it. And so, in other words, this is sort of a fictional first model based on the best available data indirectly, but I needed to be able to sort of throw quantitative models, for example, determining rates of certain processes. But do not take this as face value. So in a real vesicle, I would imagine that there would be probably a third to a half of this covered with something which is important to the cell, which is important to that vesicle, but we don't know where it is and we don't know what exactly it is.
To some extent, you're going to be dealing with the statistics of small numbers too.
Yes, absolutely, and that's a very important point actually.
That has one ATP synthase, and then you may have another one next door that has two, and it may just be stochastic as to which one it is, but they may be quite different.
In that regard, I want to sort of pass on a bit of wisdom given to me by my colleague John Olson from Neil Hunter's lab. And, he said, you think too much like a physicist. The cell makes so many of these things that if you know your vesicles are dead on arrival, it's okay. Somebody will deal with them eventually. And so, I mean, I have to sort of warn myself along with my audience here that the tendency to over-rationalize biology is a humbling experience. I mean, it's in my, some of my former collaborations, like with Petra Perome, for example, looking at the pigment organization of photosystem 1, I remember coming to her with great excitement about why a particular pattern emerges, and then it would turn out that I didn't know that phycobilisomes existed, and that whatever pattern that I thought it was actually had nothing to do with reality, because it's, you know, so essentially what I want to sort of warn all of us is that over-rationalization is probably a little bit dangerous. So with that in mind, I'm going to talk a little bit about how these models are constructed. Also consider this, how should I say it, a public service announcement. Basically what we can do for the chromatophore, perhaps we can do it for the kinds of systems that some of you are working on, particularly biohybrid systems and larger assemblies of bio-inspired pigment arrays. And of course, there's always the problem of different photosynthetic apparati, such as the ones found in plants and cyanobacteria, and so on. So long story short, we have crystal structures or structural models for the individual proteins in most cases. This has been supplemented over the last decade by AFM images of supramolecular organization. This is complemented by spectroscopy data, which turns out to be important because this gives you stoichiometry information, usually about light harvesting complexes. In this case, for example, the data shown here comes from R.V. Freiberg's lab, and people like Bob Niederman can tell us about protein association likelihoods, like if I were to grow vesicles and if I were to pass them through some torture, which proteins are likely to be found next to one another. So all of these sort of become part of the know-how, and eventually there's some mathematical transformations that you apply actually to these images, such as like area conserving maps. If you think about it, it's a little bit like geography. You can't have a map that is both area conserving and angle conserving between a plane and a spherical domain. So you have to distort something. That means that all the mappings that you do from a planar patch to a curved vesicle is actually local maps with some distortions. So that means that you have to go in there and change things by hand. The first vesicle I constructed was probably, well, it took close to a year of my time. Right now we actually automatize some of these things. So there are now hopefully less torturous ways to actually build vesicle models. So once you have done this, it turns out that you're still wrong at the end of the day because you failed to take into account a number of other features. I mean, it turns out that the LH1 reaction center dimer complexes are actually curved in nature instead of the particular geometry that we had assumed during our first study. And it turns out that the stoichiometry of the components, the pigment packing density, and the location of the components that are not imaged are all things that you can improve upon. So we have recently actually improved upon our earlier models, and lately we have also started adding, as I have mentioned before, components like the BC1 complex and HB synthase. So with that note, I'm going to sort of change gears mid-talk and cross my fingers and see if this thing works. Oh, there you go. Now, this is the show and tell part. Now, before I tell you more about the chromatophore, this is also a nod to the outreach effort that I know is happening at the Photosynthetic Antenna Research Center, which is a substantial component, and a lot of effort is going on in this direction. So we have at Urbana-Champaign have been using actually these sort of structural models, and as you see, this is running real time on my laptop, so I'm not cheating anything here. So we have been using these structural displays and demos for outreach purposes to basically school groups of any age that you can imagine. I think I've given every version of this demo from maybe preschoolers to college students, and you can tell a lot about the organization of life. Actually, you don't have to be specific about the quantum mechanics of photosynthesis, but turns out that kids are actually very interested, and many of them have heard about what a chlorophyll is, so you can tell them a little bit about what it actually looks like. It's a bunch of atoms that are put together, and you know what, these are carbon atoms, and here's a magnesium, and this thing is like an antenna. They understand antennas. You can tell them about what happens when one of them is hit by a photon. And then you can tell them a little bit about that these things are like a bunch of friends that are networking together, which is not too much of an exaggeration, actually. I mean, so excitement comes in, and you don't have to use technical terms. Excitement is shared, and excitement jumps from one group to the other. There's a narrative here, and the narrative is highly communicable. And so these demos, over the last summer, we actually started talking to high school teachers and help them develop their own narratives with the kinds of systems that they're familiar with. So this goes a little bit beyond probably necessarily the immediate domain of photosynthesis, but I would like to sort of impress upon you that there's probably some outreach potential that we can share regarding sort of computer-based displays of photosynthetic systems. And, oh, by the way, this is a very good opportunity if you have questions about structures, because before I go ahead, I can zoom into anything you want to see. And so interrupt me here now if you feel like it.
Why do you think nobody's been able to see the BC1?
Oh.
They have to be there, and do you think that they would be?
Okay, again, whatever I tell you is essentially what I have heard from Neil Hunter, John Olson, and Pu Qian. And so regarding ATP synthase, I think the answer is simple. What does an ATP synthase look like if you chop off the top mushroom? It looks exactly like an LH2. So the chances are you probably see the C rings of the ATP synthase emit a sea of LH2s. And in fact, in Bob Niederman's gel electrophoresis data, you see that there's a more likely association between the ATP synthase peptides and the LH2 peptides. So that is probably one answer to this. There is, for the ATPs, for the BC1 complexes, they actually have a very bumpy topography. So as I understand, the AFM tip probably jumps over the region that corresponds to the vicinity of the BC1 complex, prohibiting its direct imaging. Again, I repeat, this is not my expertise. I'm just relaying you whatever I have learned from my collaborators. There's a question there, yes? Equilibration times of what? Right. Okay, let's have a separation of time scales here. The assembly of the complexes probably happen seconds to minutes, I would say. I mean, they are probably delivered to the membrane, and then they are self-assembling in the membrane. This is a topic I haven't touched because, admittedly, I'm not an expert on it. But once these assemble, I will show that briefly, but these proteins induce membrane curvature by themselves. So they probably self-aggregate into domains, and then they form vesicles subsequently. Not in all species, in some. But basically, by the time this happens, your, for all intents and purposes, for the description of the excitonic behavior, your system is frozen because the time scales are separated by like 9 to 12 orders of magnitude almost. So if there are pigment protein groups like maybe alpha beta membrane dimers that contain some pigments, they may temporarily have different descriptions of the light harvesting behavior, but this will not drastically alter the light harvesting response of the system. Does that answer the question? Let us maybe talk about it offline in more detail because I don't want to get into the assembly as an extended discussion right now. So I will sort of continue. In fact, this hopefully will relate a little bit to your question. I want to acknowledge the works again of Jen Sin and Danielle Chandler. What they did is do membrane molecular dynamic simulations of light harvesting complexes, and what they observe here is very interesting, that if you take Rhodobacter spheroides light harvesting complexes, toss them on membrane, and simulate in their native environment, they curve the membrane. The LH2s start bending the membrane. I mean, of course, the accessible simulation timescales will not give you the liberty where you will actually see a vesicle assemble. This cannot be done in a full molecular dynamic simulation currently. Maybe in the future in a coarse grain description, but we are not there yet. And for the LH1 reaction center Pop-X dimers, there's actually a twist in addition to the bending, which will turn out to be very important because you will find a cylindrical array of reaction center LH1 dimers that are a result of this bending and twisting arrangement. So from left to right, the regular chromatophore sums all the parts that we do not account for. So this is from Rhodobacter spheroides. This is based on our collaboration with Neil Hunter's group. So the middle system, so this is something that Danielle Chandler had been working on with Simon Shoring, is based on the photometricum. And so this system uses actually monomeric reaction centers, and it's a planar arrangement, no curving. And to the right is again Rhodobacter spheroides, but with a mutant that doesn't have LH2s. So then all the curving behavior that you see, the reaction center dimers forming in these dimer arrays, then make these very long tubules. It's, as a mathematical curiosity, this is one of the few cases where you will see in nature a regular tiling of a membrane domain. There are very few other cases. You can see tiling patterns in viruses over finite surfaces, but regular tiles are ever very rarely encountered in biology otherwise. And again, a note to Danielle, she has actually been doing a monster simulation of the system, and she has now 10 nanoseconds of simulation time acquired on this. And this is our actually current largest simulation that is running in terms of system size. So after this gallery of systems, now I want to go back to the excitation transfer characteristics and tell you a little bit about what we do once we have the structure. So we have like two caps. One is sort of a construction worker. And when I remove that cap, I put on the cap of the physicist and ask myself, how does the system actually harvest sunlight? So how do you relate structure to function? What helps you again is the separation of pigment groups. And the modified first served formalism then gives you the rate of excitation transfer between different pigments. So you have a network of pigments that naturally divide themselves into clusters, which at the end of the day let you formulate the system in a more simplified language. Instead of having 4,000 components, right now I have a little over 100 different pigment groups. And at this point, I can now do what I couldn't do at the pigment description. At the pigment description, if you try to describe excitation dynamics in a Markovian picture, you will fail, because you will not be taking into account the quantum mechanical effect of delocalization and coherence. And it turns out that as a result of this, if you try to use the wrong formalism for the problem, you will no longer meet the resonance criteria, and the excitation transfer efficiency will actually drop significantly in such a description. But at this scale, when I'm describing the transfer of excitation between clusters of pigments, I can now successfully do this, and I can now describe the Markovian description of excitation migration between the pigment clusters rather than the individual pigments. And so at this point, then, I can compute the average excitation lifetime and quantum yield, and it turns out that in most of the arrangements, actually, the excitation lifetime is about 100 picoseconds, which gives you a light harvesting efficiency of about 90%. And there are a few other perks that you get out of this. For example, you actually observe a strong connectivity between components. So there are multiple paths to the nearest reaction center, which is one of the features that displays a sort of robustness in structure. And there is also actually significant excitation sharing between neighboring reaction centers. So basically, if excitation arrives at the reaction center, and then there's actually a significant chance of detrapping from their reaction center, there's a reasonable likelihood that that excitation will actually be recaptured by one of the neighboring reaction centers. And so then you can throw the same formulation on any kind of arrangement of pigment protein complexes. Basically, what was done for the spherical chromatophore can then be repeated for the other chromatophore models. And at the end of the day, of course, what you want to do is maybe consider a case where you consider the following processes also of charge migration and eventually ATP synthesis. And if you do the math about how much of a turnover rate such a typical chromatophore would have at one of the low light intensities that probably these poor cells have to suffer, if you do the math, it turns out that the light intensity is about less than 1% of what you would have on a bright July afternoon. A chromatophore that looks like this would be producing at a rate of about 50 ATPs per second, which is nearly half the saturation rate of a single ATP synthase. So with that note, I now want to come back to this list. So I have 4 criteria I have listed that a photosynthetic system has to worry about. 1 is improving what you do. 3 is protecting your investment, essentially. And I'll start with optimality as a design criterion. And the first case is a rather old one, but it is dear to my heart, so I'll start with this. Let's consider cyanobacterial photosystem 1. Compared to these nice cylindrical arrangements of pigments, it shows a very intricate geometry of pigments. So one thing that comes to mind immediately is this random, or is there some reason to this madness, as it were? And turns out that the pigment geometry of photosystem 1, as observed in nature, has a better quantum efficiency than a random arrangement of pigments that are located at the same locations, that are placed at the same locations. So there's a histogram of pigment arrangements, randomly reoriented chlorophylls in the photosystem 1 geometry. The only thing I don't touch here is basically the reaction center pigments. And I look at the quantum yield. And yes, it is distributed again a very narrow window that's close to 100%. But within a very narrow window, the original distribution, the original quantum yield is actually near the top of the distribution. Now, if you repeat this, for example, like the Fleming group did, you get a similar result for the distribution of site energies as opposed to the pigment orientations, and you get, again, similar results for photosystem 2, as was shown by Sergei Vasilyev and Doug Bruce. Now, turns out that this is actually slightly overstated, so let me back up. Most of this contribution actually comes from the immediate bridges that link the external chlorophyll array to the reaction center. So not all chlorophylls are created equal, some are more special than others. And naturally, it is the reaction center that is the most important, and the immediate bridging chlorophylls to them that have the greatest relevance afterwards. And after that, what you get is essentially a random cloud. And a more recent example is something I have learned from Elad Harel at Northwestern University. He has recently looked at why does LH2 have 8 or 9 subunits? And he asked this question of how you would get the best excitation transfer behavior as a function of the number of units in an LH2-like arrangement. And even though it's sort of a shallow dip here, you actually see that 8 or 9 actually gives you sort of the best arrangement. Does this actually mean something substantial? I do not know. But I think it is of note that nature probably worries about optimizing these sub-processes. And actually, let's keep in mind, excitation transfer is not a rate-limiting step for photosynthesis. There are much greater losses than the near 100% efficiency that you get for just packing closely the chlorophylls that you have in a narrow volume. But nature seems to be caring about shaving off this fraction of a percent in efficiency. And another such example is pigment density and spacing. Bob and colleagues have been writing on the evolution of photosynthesis for quite some time, and one of the things that we learn is that probobacterial systems are actually evolutionarily more primitive than their cyanobacterial and plant counterparts. And if you do the math about, well, okay, I mean, just compare how they are packing the pigments, you can do 2 comparisons. One is in terms of volume, the other in terms of the total amount of biological material that they have to gather. And one thing that you learn is that the immediate chlorophyll per amino acid ratio is actually a much better one in cyanobacterial photosystem 1. And so this is like 1/27 in photosystem 1 in cyanobacteria versus actually a comparable system would be an LH1 reaction center and maybe 4 LH2s that I believe brings the ratio to 1/40. So there's actually a better packing, not only in terms of the amino acid count per pigment, so that you're using less biomaterials to do the same task, but you do it in a smaller space, which is important because smaller space means smaller distances between pigments, greater excitation transfer rates, and in other words, a greater excitation transfer efficiency as well. Another theme that seems to be important is the use of the exciton structure and the related issue of the coherence of the excitation transfer behavior. As I have mentioned before, if you have a number of pigments which are sitting in sufficient degree of separation or enough disorder so that they don't share excitons, if you do the math with an arrangement like LH1 reaction center pigments, the transfer efficiency actually drops significantly, if not for the presence of the excitonic behavior. And recently, of course, the labs of Graham Fleming, Greg Engel, Ellen Asper-Guzik, and others have discussed the role of coherence. And it turns out that there's actually a residual coherence present even at room temperature. What I hear from Ellen Asper-Guzik is that the coherent contribution to excitation transfer efficiency is probably on the order of 5% or thereabouts. So again, the question is this intentional as part of a sort of a physics-based design rationale, or is that just a natural corollary of the close packing of pigments? A related question would be, do you see this consistently, for example, for photosystem 1 and photosystem 2? And I don't know the answer to that. And lastly, I should mention that sometimes the things that you try to optimize compete with one another. So in this case, for example, you can see the competition at the protein level or at the system level. At the protein level, you have, on the one hand, the desire to transfer excitation to the reaction center as efficiently as possible, and at the same time, you want the resulting excitation that was delivered to the reaction center causing a charge-separated state with the maximum possible efficiency. These compete because the pigments that want to be close by to the reaction center are also candidates for charge to leak out from the electron transfer chain. What this results in is what is called a cordon sanitaire, which is to say you have a 1 over R to the 6th process, which is the first start transfer of excitation into the reaction center, competing against an exponential decay, and turns out that if you do the math, you need maybe like a 20 to 30 angstrom separation from the reaction center to the cloud of pigments that are nearby. And if you do that, you can do a safe transfer into the reaction center without worrying about electron leakage out of the electron transfer chain. Again, a common theme that is found in multiple reaction centers. Now, another example of competing processes, now this time at the scale of an entire chromatophore, would be, so again, you want to do efficient excitation transfer, which requires that the pigment clusters are closely enough packed that, for example, if I'm transferring energy from LH2 to an LH2 to an LH1 to a reaction center, this is done with reasonable efficiency. Whereas I don't want them too closely packed so that I can permit quinone diffusion between these complexes. And if you ask, for example, how does the excitation transfer time and related efficiencies change as a function of the packing between the chlorophylls? It actually gives you a rather generous window over which you can maintain a high efficiency. Let me actually tell you what is on this graph. So on the x-axis is sort of an abstract expansion factor. I take the chromatophore and I ask myself, what would have happened if I sort of blow it up a little bit? No distortion to the individual pigments. I just basically ask, let me just fill stuff with lipids that separate the average distance between proteins. My efficiency will drop, of course, but I will have an easier time diffusing quinones in the system. And it turns out that you actually have a generous window over which you will still maintain a good efficiency. Now, I'm going to sort of switch gears from optimality to protecting my investment. So talking about robustness, I now want to pick something that actually sits in the middle. Thermal effects. Thermal effects turns out to be both a source of trouble and a source of profit for photosynthetic systems. they are a source of trouble because they're, well, I mean, if you have too much thermal noise, you will lose excitation transfer efficiency. But it turns out that at room temperature, light harvesting species not only actually cope with the presence of thermal disorder, but the resulting thermal broadening actually enhances the excitation transfer efficiency. One notable case, again, is photosystem 1. Both cyanobacterial and plant photosystem 1 has near 100% light harvesting efficiency, defined in terms of the quantum yield. But at room temperature, if you cool it down, the light harvesting efficiency becomes wavelength dependent and drops to lower than 50%. And in some cases, you actually get trapped in pigments that are not functionally linked to other pigment groups because you have lost the resonances that you used to have at room temperature. So the description of thermal effects, I will stay away from this. Let's just say that there are multiple methods to describe static disorder, dynamic disorder, as well as the more recent methods that describe density matrix-based formulations. But at the end of the day, there are more questions that you can ask in terms of the effect of thermal disorder on the system. For example, you can ask, what happens if I consider a thermal redistribution of my site energies in the system? And it turns out that the quantum yield of the system is actually fairly robust for a reshuffling of energies at room temperature. So this is again another sign of robustness. And another measure, again going down my list, would be the ability of the system to deal with component loss. So this brings us to graceful degradation of the system. And at a protein level, as opposed to the system level, chromatophore level, at a protein level, in photosystem 1, this is, for example, manifested by what happens to the excitation transfer network if you delete individual chlorophyls from it. And not surprisingly, except for a few central components, the deletion of most any chlorophyll from the network actually will not cause any great disruption of the excitation transfer behavior. Of course, going back to Bob's question about sort of the statistics of small numbers, if I ask a similar question at the scale of the functioning of the chromatophore, the loss of 1 ATP synthase will kill you. And so how to ask that question gracefully is not something that we have done yet, I believe. And one more side remark probably is, as they say, behind every successful chlorophyll, there's a carotenoid. So photoprotection, as we know, is a very important component. Again, as a design principle of light harvesting systems, because if a pigment doesn't have an immediate carotenoid neighbor, more often than not, it is only because it has another immediate pigment neighbor which it exotonically shares energy immediately. Otherwise, you create exotonic states that destroy the system very rapidly. And lastly, a measure of the adaptation of the system, I believe, is shown as a response to growth conditions. So we have, again, in collaboration with Neil Hunter and R.V. Freiberg, actually developed chromatophore models that correspond to different light intensity growth conditions. And the stoichiometry of the components naturally changes. And once again, I'm referring to Bob Nieder's work too, which we have not yet been able to gracefully address, but he observes a great variety of vesicle types at different stages of growth in response to the growth conditions. And so with that note, I think I'd like to sort of conclude by giving you sort of a summary in a nutshell. Basically, the light harvesting function at the scale of the entire photosynthetic apparatus integrates multiple competing sub-processes. And it does so by cooperation of hundreds of different proteins along with their thousands of pigments. And so with that note, I want to thank my collaborators again. And if you have any questions, I'm all yours. Thank you.
Okay.
Are there any questions from the group here?
The stoichiometry that I have used for a low light adapted vesicle, this is work that is not yet published. So ask me again in 2 months, I would say, after I consult my collaborators again. But so the number would be about 7 BC1 complexes to about, I believe, 15 reaction center LH1 dimers to about 80 to 90 LH2s. This is off my memory. We can talk offline and I can actually check the numbers for you, along with 1 ATP synthase.
This is actually a really important issue of getting these stoichiometries relative to correct.
Right.
It's going to be very different if you've got, say, equal number of BC1s as you have RCs and a dozen ATP synthases. And those are ultimately experimental questions that need to be narrowed down, and I don't think that.
Absolutely, and this ties actually also into another important question, which is what is the arrangement of BC1 complexes? Because I have heard some BC1 experts say that BC1 complexes form supercomplexes with reaction center LH1 complexes, and I have seen some other experts say no, that is not the case. And so, I mean, we have, of course, our neighbor Tony Crafts, and we talk to some other people. So once again, I repeat, what I show you is fiction based on consensus, specifically in terms of, for example, the placement of BC1 complexes. But this is the best answer I can manage at the moment. Yes?
So one of the slides you mentioned that economy is stable under the law of pigments.
For photosystem 1, I believe, is what I was mentioning.
Wouldn't it be more efficient to just produce plastic pigment in the first place? If the quantity
Okay. So this is a very important question, and I haven't touched this at all. Okay. So the question is this. I have a network of pigments, right? And for some reason, I picked out of the blue that quantum yield is a good measure of the efficiency of the system. Of course, that is ill-stated, because what I should really consider is quantum yield times the number of pigments that I have, or rather, the quantum yield times the overall absorption cross-section that I have. Because what is the best possible scenario for getting the maximum quantum yield? I have nothing but the reaction center. I have no peripheral antennas. Then I have the best possible quantum yield, because I will not waste time reaching the reaction center. And I will therefore have an almost 100% light harvesting efficiency. The problem with that, of course, is it's very expensive to do so. The problem, again, that we have not yet adequately addressed is that we do not yet have a cost function to describe what goes into the production of the components and what the system really optimizes for a given set of growth conditions in terms of the peripheral components. I have brushed this under the rug, so thank you for calling me on it. But so the quantum yield is a poor measure. I have admitted in the beginning that when I talk about optimality, I'll talk about a sub-process with a limited focus. So this is the reason why the quantum yield is probably not just by itself a sufficient measure of the overall performance.
Do we have any questions from the panel?
Yes.
So just to follow up on that a little bit, there's lots of interest now, especially in the case of algal systems, bioenergy, to truncate the antenna, to decrease the size of the antenna, because the systems will saturate at fairly low light intensities, and once you've saturated basically all subsequent photons are displaced. So could you, and this has been a little bit, shall I say, controversial, or there have been conflicting evidence as to how well this works to truncate the size of the antenna. Could you do this in the computer, in your system, in the sense to dial down the number of reaction centers and the number of, and sort of have a wider dynamic range that you're able to get just from growth modulation?
The short answer is yes.
Because it seems like that would be a really interesting, interesting thing to do and relevant to this sort of larger discussion of antenna size. It's really one of the most important issues facing.
Absolutely, yes. And I think, so the short again, yes, absolutely, that's very interesting. To elaborate a little bit further, there's, again, probably two different things to look at. One at the protein level, the other at the system integration level. At the protein level, especially if you're arranging sort of artificial mimics of light harvesting complexes, is of course you have to go back to the drawing board and redo the sort of the physics of the process. And let us keep in mind that photosynthetic systems have to survive a wide range of problems. They also have to, manage their life, go to school, and pay their taxes. And if you just tell them, no, I want you to just grow and harvest me energy, a lot of the machinery that, for example, fills up the surface of a chromatophore, now jumping to the system integration level, becomes unnecessary. How much of that can be truncated at the system level is probably not yet known. How much of that can be truncated at the protein level is much more easily computable. So that is probably the right point to start from.
I think if there's no questions from outside of WashU, there's no questions here. Let's thank